And we are live. Thank you for joining us for the second annual Black in Immuno Week, a week where we will celebrate, amplify, and support Black voices in immunology. Before we begin, we would like to thank our major sponsors, Immune Deficiency Foundation and Imperial College London. Don't forget to tweet about our session today using BII at the bedside, which each word capitalized for accessibility purposes. We'll have live captions and ASL interpreters during the sessions, and you can submit questions through the comment function on the platform. As a reminder, we require all audience members to be polite and respectful while in the Black in Immuno space. If we see anything offensive, your comment will be deleted immediately and will be banned from the platform. After the conclusion of the week, recorded videos will be posted on YouTube channel and transcript will be made available on our website, blackinimmuno.org, blackinimmuno.org. My name is Joël Babdor, and I'm a postdoctoral researcher at UCSF. I'll be the moderator for this session today, and I'll start right away with a huge honor to have Tonya Aaron joining us today. Tonya Aaron is a seventh-year MD PhD candidate at the Albert Einstein College of Medicine in the laboratory of David Fuchsman in the Department of Pathology. She was born and raised in Fort Lauderdale, Florida, and received her BA in biochemistry and molecular biology. Her research focuses on the effects of inflammation on the longevity of humoral immunity in the context of model antigen immunization and infection models. Thank you, Tonya, for joining us, and the floor is yours.
Thank you so much for that introduction. So let me get this screen share going. Okay, so today I'm going to be talking to you all about inflammation and how it depletes humoral immunity by limiting ASC retention in the bone marrow. So as we all know, antibody secreting cells are formed throughout the duration of the immune response. And a small population of these cells will migrate from the secondary lymphoid organ to the survival niche, which is predominantly in the bone marrow. So I'm going to show you a very basic depiction of the survival niche with the antibody secreting cells in purple, bone marrow stromal cells in green, endothelial cells in orange, and a generic hematopoietic cell in blue. So bone marrow stromal cells will secrete CXCL12 chemokine in order for ASCs to migrate from secondary lymphoid organs into the bone marrow parenchyma, where they will adhere and engage with the bone marrow stromal cells, as well as interact with hematopoietic cells that have been shown to secrete survival molecules, such as APRIL and IL-6. So recently, this term coined immunological amnesia has been shown in a variety of infection models, where whether it's in mice or looking in human serum, people have found that after severe infection with one of these pathogens, not only is pathogen-specific antibodies depleted, but what people were previously vaccinated for, even decades prior, have also been depleted. So these pathogens have different mechanisms that will either prohibit the ASCs from interacting with the bone marrow stromal cells, that can directly infect bone marrow ASCs, or deplete hematopoietic cell populations that contribute to their survival. But one thing that's less studied is the role of systemic inflammation and how that plays a role in immunological amnesia, because all of these pathogens induce severe inflammation. So when we look to the literature, we found in the study from the Gray Lab that when mice are treated with recombinant TNF-alpha, there's a depletion of bone marrow plasma cells, specifically in the bone marrow and not in the spleen. But it was unclear from this paper whether or not this decrease was due to mobilization or if TNF-alpha was inducing cell death. So our first question was, can recombinant TNF-alpha deplete the duration of humoral immunity using model antigen? So what I did was I adoptively transferred B cells with high affinity BCR to a model antigen, nitrophenol. And 6 weeks post immunization and adoptive transfer, I began to collect serum for antigen-specific IgG titers in serum. And I treated the mice with PBS, or recombinant TNF-alpha, for 3 consecutive days, and then collected serum at days 7, 14, and 30 post TNF-alpha or PBS treatment. And what you can see here is that with the PBS treated group in black and the TNF-alpha treated group in blue, that there's a significant rapid depletion of MP-specific IgG in the serum with TNF-alpha treatment, indicating that TNF-alpha can recapitulate this depletion of humoral immunity, which was seen in the various infection models. Next, we wanted to see if TNF-alpha was inducing ASC apoptosis or mobilization in the bone marrow. In order to track our ASCs, I intrativally labeled the bone marrow with CFSE, and you can see that here in the injected tibia, with about 15% of the ASCs being labeled. And then after those three treatments that I previously showed you, that population is significantly diminished. And what you can see here is that when you look in the injected bone in PBS and TNF-alpha treated mice, there's a significant decrease in our labeled ASCs. However, you begin to see an increase in our labeled ASCs in the spleen, indicating their dissemination with TNF-alpha treatment. However, we did not see the same trend in the contralateral tibia, indicating that TNF-alpha may be inhibiting ASC re-engraftment once they've been mobilized. So in order to test this question, we did an adoptive transfer experiment where recipient mice are treated with PBS or recombinant TNF-alpha twice a week for 3 weeks. And after the first week of treatment, I adoptively transferred YFP positive ASCs into our recipient mice. And at 14 days post-adoptive transfer, I looked for those YFP positive ASCs in the bone marrow and the spleen. And what you can see here is that the YFP ASC engraftment into the bone marrow of our recipient mice was significantly inhibited with TNF-alpha treatment. However, there was no effect on splenic engraftment. Then we want to see if we could rescue ASC engraftment by blocking TNF-alpha signaling in the context of inflammation. So with our recipient mice, we treated them with anti-TNF-alpha monoclonal antibody or isotype control antibody for 3 weeks. Within the first week of treatment, we injected these mice with alum adjuvant to induce inflammation so that we can specifically target TNF-alpha in the context of inflammation. And again, we adoptively transferred our YFP ASCs and looked for their engraftment at day 14 post-adoptive transfer. And what you can see here is with anti-TNF-alpha treatment, we significantly improved the engraftment of our YFP ASCs in our recipient mice, as well as improving engraftment in the spleen, leading us to believe that not only is engraftment improved by blocking TNF-alpha, but we are improving the overall survival, and bone marrow engraftment is necessary to ASC survival. In order to look at this, we used our mice that have a YFP on a BLIMP1 promoter to identify our plasma cells. And I gated on mature plasma cells that are CD138 high, B220 low, and CD3 negative in the bone marrow, spleen, and blood. And this is just indicating that they're of similar maturation. But what you can see here is that in the bone marrow with TNF-alpha treatment, there's no significant change in survival. It is only when these cells have been mobilized from the bone marrow in which you see a decrease in pro-survival factor BCL2 expression, especially when those cells are in the blood. So if TNF-alpha is inducing the mobilization of ASCs from the bone marrow and preventing them from getting back into the bone marrow, it's having a drastic effect on their survival. Next, we wanted to determine which cellular compartment was regulating ASC retention in the bone marrow. So in order to do that, we generated bone marrow chimeras where our recipient mice are irradiated and we use the bone marrow from our donor mice to reconstitute the hematopoietic compartment of our recipient mice. And one thing that's cool about ASCs is that in the bone marrow, there's a significant population that's terminally differentiated, and they're no longer proliferative. So a portion of these cells will remain after radiation. So we can compare and analyze our donor and radiation-resistant ASCs. But more on that a little bit later. So what I want you to do is think of this recipient population as our non-hematopoietic cells and the donor population as our hematopoietic cells. And what you can see is when our non-hematopoietic cells express TNF-alpha receptor, because in this chimera, both TNF-alpha receptors are knocked out, receptor 1 and 2. So when the non-hematopoietic cells express TNF-alpha receptors, we get an increase in ASCs in the blood with TNF-alpha treatment. However, when those receptors are absent in our non-hematopoietic cell population, we lose this effect. And when we look in the bone marrow, we see that bone marrow ASC numbers are significantly reduced with TNF-alpha treatment when the non-hematopoietic cells express TNF-alpha receptor, and lose that effect when receptor expression is absent, leading us to conclude that non-hematopoietic cells are regulating ASC retention in the bone marrow. And we don't see any effect in the spleen. Next, we want to identify which receptor was responsible. So we generated chimeras with single receptor knockouts. And what you can see here is with the absence of receptor 2 in both hematopoietic and non-hematopoietic compartments, we still see an increase in ASCs in the blood with TNF-alpha treatment. And we also see this effect of a decrease in ASCs in the bone marrow in all of our chimeras, indicating that receptor 2 expression is not necessary for ASC retention in the bone marrow. And in order to confirm that receptor 1 was regulating ASC retention in the bone marrow, we generated receptor 1 chimeras. And what you can see is that we lose our increase of ASCs in the blood with TNF-alpha treatment when receptor 1 expression is absent in our non-hematopoietic cells. And we also lose that decrease in ASCs in the bone marrow when receptor 1 is absent in our non-hematopoietic cells. And it's to confirm that the ASCs are being mobilized with TNF-alpha receptor 1 signaling, we did that dissemination experiment that I showed you earlier where I labeled ASCs in the bone marrow. And you can see that when receptor 1 is absent in the non-hematopoietic compartment, we do not see this increase in dissemination, leading us to conclude that receptor 1 signaling in non-hematopoietic cells regulates ASC retention in the bone marrow. So what is receptor 1 doing? How is it modulating ASC motility and egress? Well, previously in our lab, with our multiple myeloma studies, a postdoc in our lab showed that CD138 expression on ASCs can be modulated. And as the receptor expression goes down, these myeloma cells become more motile and more readily disseminate from their primary site and disseminate into other locations in the body. So we wanted to see if TNF-alpha can decrease CD138 expression on ASCs. So when I treated mice, wild-type mice with TNF-alpha, I do in fact see a decrease in CD138 expression. And I also saw the effect in receptor 2 knockout mice in bone marrow ASCs. However, that effect was lost in receptor 1 knockout bone marrow ASCs. And next, we wanted to see if this effect was cell intrinsic or extrinsic. So we went back to our chimera data. And I remember I told you that there's going to be a population of ASCs that remain in our recipient mice with chimeras. So when our non-hematopoietic cells are wild type, we see a decrease in CD138 expression, regardless of TNF-alpha receptor expression in the ASCs, in both donor and recipient populations. However, when receptor expression is absent, we lose that decrease in CD138 expression, leading us to believe that TNF-alpha is causing a decrease in CD138 expression in bone marrow ASCs in a cell extrinsic fashion. So one thing to note is we see this decrease in CD138 within 6 hours. So we're able to use intravital imaging to see the effect of TNF-alpha on ASCs in the bone marrow in real time. So here we have mice with the vasculature labeled in tomato and our YFP positive ASCs. And we can look at these cells pre and post TNF-alpha treatment in the same location. And what you can see is with TNF-alpha treatment, there's an increase in track velocity and displacement velocity, indicating that TNF-alpha is rapidly inducing their motility. And a postdoc in our lab was also able to generate this figure in which you're able to assess the distance between ASCs and the vasculature pre and post treatment. So as the cells become more purple, that means they're getting closer to the vasculature, and if they're closer to red, they're further away from the vasculature. And you can see here in this calculation that the distance between ASCs and the vessels decreases with TNF-alpha treatment. And finally, we wanted to look at how TNF-alpha receptor 1 signaling was affecting this chemokine receptor axis, because this is a well-studied mechanism of retention for not only ASCs in the bone marrow, but hematopoietic cells in general. So one thing to keep in mind is that when CXCL12, active CXCL12 is high in solution, CXCR4 expression goes down on ASCs and vice versa. So when CXCL12 is low, we expect CXCR4 expression to go up. When we look at immature and mature ASCs in the bone marrow, we see that TNF-alpha treatment increases CXCR4 expression on both populations. So then we look to see what the CXCL12 levels were in bone marrow serum. And to our surprise, we saw that these CXCL12 levels actually increased with TNF-alpha treatment. And then when we want to see if we would see the same effect in TNF-alpha receptor 1 knockout mice with TNF-alpha treatment, we lost this increase in CXCL12 secretion. So that led us to conclude that this axis is being disrupted in the context of TNF-alpha mediated inflammation. So I know there was a lot that I went through in this short amount of time, so I'd like to summarize the findings that we've had. Recombinant TNF-alpha can deplete humoral immunity, similar to what was seen in the pathogen models. Bone marrow engraftment is critical for ASC survival. TNF-alpha receptor 1 signaling in non-hematopoietic cells regulates ASC retention in the bone marrow. TNF-alpha signaling rapidly mobilizes ASCs in the bone marrow via CD138 modulation. And TNF-alpha receptor 1 signaling enhances CXCL12 cleavage while disrupting the CXCR4-CXCL12 retention axis. And with that, I'd like to thank you for your time and attention, and I'm happy to answer any questions that you may have.
Thank you, Tonya Aaron, for sharing this amazing investigation that you've performed. And we're going to take questions from the audience, and I can see one already popped up. So we have a question from Catherine Mayer Barber asking, do you think TNF-alpha receptor mechanisms in non-hematopoietic cells could play a role in emergency hematopoiesis in other bone marrow resident cells?
That's a great question. So I'm not too familiar on emergent I'm sorry, what? Let me read the question. Emergent hematopoiesis in other bone marrow resident cells. That's a good question. So we've seen this effect in like bone marrow resident macrophages, if that's what you're referring to as well. That's something that we definitely need to investigate to look at the different stromal cell populations within the bone marrow, as well as CD169 macrophages. That's something that we're definitely doing right now, especially trying to do like Cre-Floxed receptor 1 models to really locate which population is regulating that. Great question.
Thank you for the question, and thank you for the answer. We have another question from another member of the audience, Rachel Gernstein, asks, what is the evidence that this change impairs antibody secretion, especially functional consequence? And nice talk.
So impairs, okay, so that's, I see, that's a great question. So I guess what we're showing is that because the antibody titers are going down, the cells that make them are no longer present. But it would be nice to do like an in vitro study and treat cells with TNF alpha or not, and then take that supernatant and see how their ability to secrete antibody, if it is affected. But yeah, that's a great idea. That's something that we can definitely do.
We have a comment from Donald Palmer saying interesting talk, and a question: could you speculate on how TNF alpha impact bone marrow stromal cells?
Yeah, I mean that's the that's a big question, especially since my model is very acute. So like, how is inflammation affecting the stromal compartment over time in chronic inflammatory settings? That's definitely something I want to do moving forward in my career. But I think maybe desensitization to inflammation. How are these different populations changing? Because I know with inflammation, you can kind of, from the hematopoietic standpoint, you can kind of shift towards granulocyte generation. So it's very interesting to see how these stromal cell populations change or are desensitized to inflammation over time.
Awesome. We have another question from Adam Savage saying, thank you, this is very interesting. And the question is it known whether TNF-mediated disease like rheumatoid arthritis or IBD show similar effects on plasma cells, or whether anti-TNF treatment in human disease impact plasma cells?
Great questions. So for diseases like rheumatoid arthritis and IBD, you do see more plasma cells in circulation. Now, whether that is because of this retention mechanism has been disrupted, or whether there's just more plasma cells in these people, so the niche has reached capacity and now they're just in circulation, is something to investigate. However, when people have treated with just anti-TNF alpha, there's actually been like this rebound effect, and it's made disease worse. So now studies are looking to target specific receptors instead of just targeting the soluble TNF alpha. So I think that this study showing like this specific receptor is kind of regulating the retention is good, hopefully.
Awesome. Thank you so much, Tonya Aaron, for this fantastic talk and for answering these questions. We're going to move now to the next speaker for this series. And thank you again, Tonya. I'll now welcome Devon Eddins. Devon is a James T. Laney Doctoral Fellow in the Laboratory of Ediver Ghost at the Lowen Center for Human Immunology, Emory University School of Medicine in Atlanta. Devon's studies centered on the laboratory's goal of understanding tissue-resident versus tissue-recruited immune lineage and responses, with emphasis on innate immune regulation mediated by tissue-resident macrophages in antiviral immunity. To this end, Devon's recent studies have employed multi-omic single-cell technologies, including high-dimensional 30-plus parameter flow cytometry and virus-inclusive single-cell RNA-seq to study immune responses in hospitalized COVID patients requiring ICU admission and mechanical ventilator support. Thank you so much, Devon, for being here, and we're very excited for your talk. The floor is yours.
Thank you, Joel, for that very kind introduction, and thank you all for being here. And I'm very excited today to tell you a little bit about some of our studies that we've been doing in our cohort of Black patients, actually, specifically here in the Metro Atlanta area. And I'm first going to begin with arguably one of the most important slides. of the talk, and that is the acknowledgements, because it takes a whole team of people in order to facilitate these studies. So a big shout out to all of our group members, and along with our collaborators here at Emory University and also at Genentech for all of their hard work in making this possible. Also a special shout out to my committee members who have been very helpful in giving feedback and direction for the studies. But most importantly, I want to thank all of the patients and their families, along with the other clinical facing personnel that aren't listed here, for their dedication and commitment, and because without them, none of this would be possible. So really want to start off by thanking all of these people. And with that, there's a point that I really don't have to belabor much, and that's the COVID-19 pandemic. This has been our reality for almost going on 2 years now, and it has been quite substantial in every way that you can imagine. And just to throw some numbers that are semi-up-to-date as of Monday, there have been more than 219 million cases of COVID-19 disease and over 4.5 million deaths worldwide. And when we look at the United States alone, over 47 million of these cases, which is over 21% of total cases, have been here within the United States, and approximately 762,000 deaths, which means the US accounts for almost 17% of total deaths worldwide. And here within the United States, community of color are disproportionately affected by severe COVID-19 disease and COVID-19 mortality. And even though communities of color are disproportionately affected by disease, they are also underrepresented in a lot of the major studies that are using multi-omic approaches to understand immunity and pathogenesis in COVID-19. So to that end, we wanted to specifically focus on a cohort of African and African American patients here in the metro Atlanta area. And so we had a total of 35 patients that we were able to enroll in this study, and they are shown here in this table. So we had 18 patients that were seen in the ICU and had an NIH score of critical disease, which we will refer to as severe throughout the study. And all of these patients had acute respiratory distress syndrome and were on a mechanical ventilator in the ICU. We also had a subset of patients that had mild acute illness that were seen in the outpatient acute respiratory clinic here at Emory, and then also healthy controls to compare those two. And from these patients, our severe patients, we would get airway samples from, so endotracheal aspirate samples from their tracheostomy or where they were intubated, or the ET tube. And we would also get matching blood samples at the same time that were collected together. From the mild patients, we would also get blood samples and non-induced sputum at the same time. And then from the healthy controls, we would just get peripheral blood samples. And then we would take these samples and we would employ our multi-omic systems immunology approach in the lab, which includes SARS-CoV-2 virus-inclusive single-cell RNA sequencing with 89 antibody-derived tags or antibody sequencing, 30-parameter flow cytometry, and also 21-plex protein analysis, including assays to measure myeloperoxidase. And then we would do the integrative analyses from all of these data sets to understand better the features of immunity and pathogenesis in this cohort. And so one of the first things that we noticed that was very striking was the neutrophilia, specifically within the airways of the samples. So immune profiles in the upper airways were very neutrophilic, as you can see here in the representative gating strategy from the flow cytometry data. So taking viable single cells, you see that the majority of the cells in our sampling are CD45 positive leukocytes, and of those, the granulocytes, namely neutrophils, are the most abundant cell type. And though a lot of the early studies in peripheral blood had highlighted the neutrophil to lymphocyte ratio as being important and a potential prognostic marker for disease Early BALF and ETA studies were less clear and more heterogeneity. There was much more heterogeneity. So we didn't really have an appreciation for this pronounced neutrophilia that we see is very consistent within our cohort of Black patients. This is also reflected in the circulation, as has been previously described, and also another common feature that's seen in COVID ARDS, the decrease in natural killer cells, which has been described from other groups. And so since we had our high-dimensional flow cytometry data set, we were able to now really specifically interrogate more the phenotype of the neutrophils and the matching blood and airway samples from these patients to better understand what neutrophils are doing and how they're contributing to disease. And so within our high-dimensional flow cytometry data, we were able to identify a hyperinflammatory phenotype of neutrophils once they got into the airways. So pulmonary neutrophils, we had extracellular CD63 or LAMP3 along with extracellular neutrophil elastase in the panel. And we can use these markers to help us identify neutrophil degranulation. So when you look in the blood of the severe patients versus the matching airway samples, you can really appreciate a marked upregulation of both CD63 and also a neutrophil elastase, which gives us a lot of evidence of primary granule release is exacerbated once neutrophils are recruited from the blood to the lungs in severe patients. And we also were able to measure cytokines that are implicated in cytokine storm in patients and the total cellular compartment of the lung via the mesoscale data, but also having intracellular staining in our flow cytometry panel. We're also able to interrogate specifically the neutrophilic contribution to cytokine storm for these markers as well. So here I'm showing IL-8 first, and as you can see in the mesoscale data, that IL-8 increases in a stepwise manner with disease severity. And you can also start to appreciate an uncoupling of the systemic versus local mucosal responses, where you see a very potent and pronounced increase in the airways compared to the circulation. And so when we drill down specifically onto the neutrophils, you can see that neutrophils in the blood aren't producing very much IL-8, but however, when they're recruited to the airway, this production of IL-8 is increased substantially. The same is also true for IL-1 beta, where you could see more of a bimodal distribution in the blood, where you have some neutrophils that are clearly negative, but virtually all neutrophils, once they're recruited to the airways, are expressing IL-1 or have IL-1 beta. And then a similar phenomenon for IL-6 in the blood, and then once they get to the airway, they are expressing IL-6. So now we're starting to appreciate a hyperinflammatory phenotype where we have neutrophils that are increasing degranulation once they're recruited to the airways, and they're also increasing the amount of cytokines, namely IL-8, IL-1 beta, and IL-6, that they are able to secrete into the lung milieu. And then we wanted to better understand, well, what's happening to these neutrophils from a transcriptional standpoint. So we first wanted to see if we could identify in the blood the subset of circulating neutrophils that are likely recruited to the lung. And we were able to do this using receptor ligand and interaction analyses, which identified the CXCL8 or IL-8 and CXCR2 IL-8 receptor signaling axis as the primary recruitment axis for lungs or for neutrophils to the lung. And interestingly, we found only one subset of neutrophils in the blood, namely a subset of neutrophils that express high levels of the alarmin S100A12 and interferon-stimulated genes, specifically the IFIT-M proteins. Here we focus on IFIT-M2, that express CXCR2 were being recruited to the lungs. And so this mature population of neutrophils, we wanted to understand when they're being recruited to the lung, what happens. So here we use SCVilo to perform trajectory analyses of the neutrophils once they infiltrate the lung. And as you can see here, we really identify two distinct trajectories that we just named trajectory 1 and 2 for neutrophils once they get to the airway. And when you look at the beginning of the trajectory and the velocity plots, you can see S100A12 and CXCR2 at the beginning of the trajectory, further supporting this is the subset that's been pulling from the blood. And as they differentiate in the airways, we notice a marked upregulation of CCL3 and 4, which can recruit more monocytes and push monocytes and macrophages in the airways to a pro-inflammatory phenotype, and also induction of more ISGs, namely here we include IFI30. And when we perform pathway and process enrichment analyses, we could see pronounced features for neutrophil activation and degranulation, along with platelet activation and aggregation, response to interferon gamma, and just innate and adaptive immune regulation overall. So our next question that we wanted to ask was, what are the ligands that are present in the airways that contribute to this transcriptional reprogramming, allowing neutrophils to acquire a hyperinflammatory state in the airways? So TNF, when we did these analyses, TNF and IL-1 beta were identified as the drivers for the inflammatory reprogramming of the lung-recruited neutrophils. And so here again, I'm just showing you the mesoscale data that TNF is increased in a stepwise manner with disease severity, along with IL-1 beta, which you've already seen. And here you can see in analyses that are performed in NicheNet, which uses the receptor ligand interactions, but also queries in the host transcriptome intact signaling pathways, you can really appreciate how TNF and IL-1 beta have broad regulatory potential on genes that are upregulated as neutrophils acquire a hyperinflammatory state, including the TNF superfamily genes and NF-kappa B signaling. And then also one thing that was really interesting, and as you'll remember from Tonya's talk, BCL2 was being induced from TNF in the airways, which is known to regulate neutrophil survival. And then looking at the cell subsets in the airways, which are producing these ligands, TNF was exclusively being produced by T cells, and IL-1 beta by transcript we see in the myeloid populations, but we also showed by protein and the intracellular staining that neutrophils are also producing a lot of IL-1 beta in the airways. So with that, I would like to conclude some of the findings that we have. And one thing that we were able to identify is this pronounced transcriptional reprogramming of mature IFIT-M2 positive neutrophils that infiltrate the lung from the circulation via the CXCL8, CXCR2 axis. And then once these neutrophils infiltrate the lung, pulmonary IL-1 beta and TNF promote a transcriptional reprogramming that induces the production of cytokines implicated in cytokine storm, along with increased neutrophil degranulation and release of neutrophil elastase and myeloperoxidase, and also increasing their survival in the airways as well. So they have acquired this hyperinflammatory state, secreting a lot of cytokines and potent proteases that are known to lead to epithelial damage and lung pathology in COVID-19 disease. And so with that, I would like to thank you all for your time and attention, and with remaining time, take any questions that you may have.
Thank you so much, Devon, for this fantastic talk. I am very impressed by this fantastic work that you've done, and the fact that this is a COVID study with Black patients studied by a Black researcher is really making me really happy. I wanna take a few questions as we're running a little bit short on time. I think we have a comment from Wesley Eddins that is very proud and saying that you're doing a great job. We have another comment from Kristen Marrow saying great talk. And let's see, I don't see questions. Maybe we have time for one or two. So let's see who shoots first. But yeah, I want to say the way that you have this multi-omic strategies to investigate is really impressive. And yeah, you mentioned that there is a whole team working on this sort of efforts. And yeah, do you want to comment a word on how this teamwork has been going on?
Yeah, it's very it's been very great working with an entire team of both the clinical facing staff. It's been a massive effort to recruit and consent patients to get them on board in the study. We're so very grateful and thankful for their willingness to participate, because without them we couldn't do this. And then having a team of people to really help, because the airway samples would have to come to the lab, and it would have to go into BSL-3 for processing, and then the blood samples into BSL-2 plus containment. So it's a lot of time spent in containment and a lot of hard work to do all these assays. And we're also very grateful to the NIH who provided the funding for this study, so we could really look at this and interrogate these dynamics within this unique cohort.
Amazing. We have a few questions. So we have Nadina saying, a really interesting talk. Did you look at how neutrophil level activation correlate with viral load?
Yeah, that's a fantastic question. And we did. So sorry, I did not include the data in the talk, but when we measure viral load and comparing our mild acute patients and our severe patients that are in the ICU, we actually see that viral load is decreased in the patients that are in the intensive care unit as compared to those that are seen in the outpatient clinic. So we don't have out of control viral replication when the patients are in the ICU. So we argue that at this point, it's important to really understand what the immune response is doing, and possibly the immunopathy that's going on more so than out of control viral replication in the airways.
Awesome. I'll take the last question. We have Mark Jenkins asking, Casanova reported that people with severe COVID have autoantibodies that neutralize type 1 interferon. Do you have evidence for that in your study?
Yeah, so we haven't looked specifically at autoantibodies in this patient cohort. We do have some studies that are coming up to better understand the serology from the blood of these patients, and also understanding the antibodies that are present in the respiratory secretions. However, we don't have any data on that or cannot comment on that at this time.
All right. Thank you so much, Devon Eddins, for this fantastic talk. And we'll move to the next speaker now. I am excited to welcome Dr. Oladipo Kolawole. And so Dr. Oladipo Kolawole is an international fellow in the ICMR IUSTRC with a PhD in medical virology and immunology from Ladoke Onkitolo University of Technology. Presently, he holds a faculty position at the Department of Microbiology at Ekele University in Nigeria. He focuses on vaccines and diagnostic kits development for neglected tropical diseases. And we should have him join soon. Let me see. It seems like we have a small technical difficulty. Let's see. I think we're gonna move to the next speaker and see if we come back to Dr. Oladipo Kolawole later. All right, let's see. Do we have Dr. Makeda Robinson in backstage? Can have Dr. Makeda Robinson to join the stage? I think
I'm here. Yes.
Oh, all right. Yes. Awesome. Sorry. So we'll go to Dr. Makeda Robinson. That I'm also excited to have here. Dr. Makeda Robinson is from Long Beach, California. She earned her MD and PhD from Stanford University School of Medicine and is currently a postdoctoral infectious disease specialist at the university. She's a recipient of Howard Hughes Medical Institute Fellowship and is a Jackie Robinson Foundation Scholar. Her current research focuses on understanding virus host dynamics in the setting of emerging viral pathogens, which has led her to pursue translational approaches to diagnose and treat novel infections. In response to the ongoing COVID-19 pandemic, Dr. Robinson has developed a research project centered on better understanding age-dependent differences in immune responses to infection to identify key windows for treatment and predict disease progression. In parallel, she is working to spread evidence-based information about COVID-19 and has commented on current issues surrounding the pandemic in multiple networks, including on a panel that we had the chance to host a few months back. Thank you, Dr. Makeda Robinson, for being here today, and the floor is yours.
Well, thank you so much, Joel, for that warm introduction. And I first just wanted to thank the founders of Black and Immuno and the organizers of this week for the opportunity to speak. It's really an honor. I wanted to talk a bit about my evolution as an infectious disease specialist over the last couple of years during the COVID-19 pandemic, so I could share a little bit of my journey to becoming a physician scientist as well as some of my research interests. And so, as you mentioned, so I had a non-traditional path to becoming an MD PhD. So I completed all of my clinical training and my infectious disease specialization. And then I started graduate school. So I started my PhD in 2015. And right around the time that I was beginning to wrap up and put together my defense, a new novel virus had popped up into the sort of general knowledge. And actually, the day before my PhD defense was sort of the first confirmed case of COVID-19 in the US. And as you can see from the title of my defense, the roles of virus-host interactions in the pathogenesis of dengue and Zika viral infections, the emergence of COVID-19 was something that was very interesting to me. And so as I was writing my dissertation, multiple new announcements began to come up about this virus. The World Health Organization declared a global pandemic on March 11th. And then for us in California was the first shelter-in-place order that was put in place in California. So during the shelter-in-place ordinance, while many labs closed, some of them still opened, of course, so they could work on SARS-CoV-2, but many of us had additional time on our hands that we didn't have before, because many of the things we would do were closed, restaurants were closed, we couldn't travel as much, and I found myself spending a lot of that time reading as much as possible about this virus and trying to understand how it was impacting all of our lives. And through that, I really came to a sense that while as scientists, we were able to really read all of this information and gain knowledge and understand the reasons why we were having to have shelter-in-place ordinances and really change our lives in a dramatic way, that knowledge wasn't always being disseminated into the general population in a way that was understandable. And so during that time, I decided to kind of add my voice to that and started a YouTube channel called Our New Viral World. And I also started sort of writing about topics that I thought were interesting. And then also, I think for many of us as Black scientists during that time, seeing, as Devon mentioned, the sort of increase in morbidity and mortality rates from COVID-19 in Black and Brown communities, I felt like it was really important for us to begin to sort of tailor some of the discussions that were being had to those populations. And I was able to give a talk to a group called Black Women in Tech, which is sort of centered in the Bay Area, but also internationally, to sort of share some of that knowledge with them. And that actually was a really great experience because it helped me to understand a bit about the questions that people ask that aren't in the sciences, that it wasn't sort of a scientific talk, but people really had questions that they were concerned about, and finding ways to answer those and have people feel as though you were listening to them was really important. And I also felt like modeling those behaviors that we were talking about, so masking and distancing, as well as getting vaccinations, that sort of modeling that behavior was really important because so many things in our life were changing, and some people were not able to understand as easily why they were important. Okay, So now getting a bit back into the science. So post shelter in place, I began to start asking some questions about COVID-19 pathogenesis after doing some of the reading I had done during that period of time. And one question that I've been interested in general, so from my graduate school education as well as now in my postdoc, is what are the cellular and molecular determinants which drive the pathogenesis of severe viral emerging infections? And a pattern I felt began to emerge with COVID-19 infections that I had also seen with another virus I had studied, dengue, where there were a large number of asymptomatic or mild cases where patients would be infected and within a few days would not feel well, but would more or less recover. But there were another subset of patients that would go on to develop severe disease. And while we had some sense of what the risk factors were for that, comorbidities such as heart disease or obesity, the immune responses that triggered this difference in immune response remained unknown. And what I had the privilege of doing in my PhD, which was generating cohorts. I worked to generate a cohort of dengue patients in Colombia, and also use publicly available data in order to make predictions about who would progress to severe disease and who would have a mild course. And I did that using a systems immunology approach, and so using two single cell approaches. The first of which was mass cytometry, or CyTOF, which is a proteomics approach, and the other was single cell RNA-seq. So this is a map I remember looking up during the pandemic that I found really interesting. This map was initially created by Dr. Fauci during the HIV epidemic, where there was just one virus, but it's been updated throughout the years to include many of the new emerging and re-emerging infections that we have been dealing with. And as an infectious disease specialist, these had popped up in my mind throughout my career. So I had studied specifically dengue and Zika, and the first year or two of my PhD was the Zika virus outbreak. I also did my clinical fellowship during the Ebola epidemic. And so I was very interested in beginning to apply some of the things that I had learned in the past to this new virus. And so Devon sort of outlined this quite well, but I'm sure as many of you know, the cases of COVID-19 have continued to increase despite the availability of vaccines, with over 250 million cases and 5 million deaths. But something that I found really interesting early on was that there was this clear age-dependent difference in both transmission and severity of COVID-19. And this Nature Medicine paper came out in 2020, demonstrating that young children and adolescents were less susceptible to infection, and also when they became infected, had fewer clinical signs or symptoms. And this difference really had no geographic target. So it was in geographically heterogeneous populations, this pattern continued to emerge. And around the same time, many groups were beginning to create COVID-19 cohorts because it was a global pandemic, and so there were many COVID patients throughout the world. And one that I found very interesting was one that had both adult and pediatric patients with mild and severe disease. So the multi-system inflammatory syndrome in children, which is rare, which allowed them to look at multiple different disease severities of COVID-19 patients. And what I found interesting here is that when they looked between children and adults, what they found is that adults actually had a fairly robust adaptive immune response, at least slightly significantly increased from that of children. And so you can see here that the neutralizing antibody titers were slightly increased in adults, as well as there was an increased response of activated T cells after stimulation with spike protein. So given that, I was really interested in understanding how were children able to deal with the virus in a way where they were not developing these severe diseases. And so I turned to look at potentially innate immune responses that might be important. And several papers had come out looking at a very specific subtype of myeloid cells called myeloid-derived suppressor cells. And they found that in severe COVID patients and fatal COVID patients, there was an increased number of these cells compared to healthy controls or mild patients in the blood. Now, these myeloid-derived suppressor cells have multiple different functions, which is why I find them so interesting. They're able to inhibit antigen presentation and on dendritic cells, reduce cell cytotoxicity in NK cells on the sort of innate immune side, but they're also able to suppress adoptive immune responses by reducing T cell activation in CD4 and CD8 positive T cells and increasing regulatory T cell activation. And these cells actually are able to survive longer in the presence of TNF alpha, which we know is one of the cytokines that's increased along with IL-6 and is associated with severe COVID-19. And so this combination of sort of early preliminary data really helped me to formulate a hypothesis, which is that early innate responses are crucial for the initial response to SARS-CoV-2 infection, and that these immature myeloid cell subtypes may play a role in age-dependent differences in disease outcomes. And so here, I hypothesized that children may have this early, robust innate immune response, where adults have a less robust response early on that allows the virus to sort of take hold and leads to downstream cytokine storm and worse outcomes. And so then fortuitously, I talked to an incredible mentor of mine, Dr. Maldonado, who has been doing a lot of amazing science communication work around the COVID-19 pandemic, both in adults and children. And they had been developing a household transmission study looking at COVID-19 within households. And so here the workflow basically entails the identification of a patient within a household that has a positive nasal swab QRT-PCR test. And then once we identify that person, we enroll them as well as all of the people in their household, and we obtain blood samples that we isolate PBMCs from. And we also monitor those patients for the next month, where they take daily nasal swabs to identify new transmission, as well as keep a symptom diary so we can identify which symptoms occur and the kinetics of that. We do that for 30 days, and then we draw longitudinal samples, both at 30 days and 90 days, so that we can look at the the duration of the immune response and how that shifts over time. And within that, what was really interesting is that as I began to work on this, more questions began to emerge and more potential collaborations were identified where we were thinking more about many other questions that we could ask with this cohort. So one of those questions was whether or not COVID-19 can increase the risk of diabetes and how it does that. We're also now recruiting a cohort of breakthrough infections, so patients who have already been infected and are now being vaccinated, and then are diagnosed with a COVID-19 diagnosis after vaccination. We can look at longitudinal effects because we are obtaining these blood samples both 30 days and 90 days after, and we're actually have some funding to extend that beyond 3 months. And then we also have been starting to recruit pregnant women into our cohort, so we can begin to look at this underrepresented population that often doesn't get as much research performed. And so with that, I initially generated a CyTOF panel to begin to immunophenotype the immune responses in both adults and children within households, and really wanted to delve deeper into this myeloid compartment to look at this specific subset of cells, these MDSCs, which within that subset has 2 different subsets, both a granulocytic and monocytic morphology, and to look to see how these responses may change with age, and also have some functional markers here so that we can also look at their cytokine secretion. And I also found another incredible mentor, Dr. Catherine Blish at Stanford, who has been supporting me on this project. And so I've really feel like these last 2 years have allowed me to also really grow as a scientist. And so lastly, I just wanted to talk about some lessons that I've learned through this process. So one is to add your voice to the conversation. If you have something to say, we're scientists who are really becoming experts in our field, and being able to share that with people, I think, is very meaningful, especially as Black scientists. Do things that make you feel uncomfortable and stretch yourself whenever you can. So helping to basically spearhead this project really allowed me to grow both as a scientist as well as a leader of a project, and to begin to ask myself the types of questions that I would need to begin my own research program in the future. Always be open to learning and gaining new skills. So I think for many of us during this time, some of us learned a lot of computational skills, and that was something that I also did, because I think, we had this time and space to really do more computational work and to find joy in the challenges. So I know this year has been really challenging for almost everyone in some way, and finding ways to learn from those and grow, I think, is really important. And so with that, I took you through my stages of evolution through these past couple years. The new normal of becoming a PhD, my sort of entry into science communication, asking new questions about an interesting pathogen. And then I hope that my fourth stage will be discovery and translation. I'm really passionate about finding ways that we can use science to impact human health, and I'm excited to see where that goes. And so with that, I just wanted to really thank all the people that have helped me with this project. Catherine Blish and everyone in the Blish Lab has been just a really incredible group to work with, and Castanedo, that's going to be working with me on this project. Dr. Balni Maldonado and Jonathan Altamirano, and my previous PhD mentor, Shreet Anav, and my funding sources. With that, I will take your questions.
Thank you so much, Dr. Makeda Robinson. This was a fantastic and exciting talk, very topical subject, and a cohort that I'm sure is going to bring a lot of new, interesting discoveries. I'll take some questions from the audience. And let me check. Okay, it seems like we don't have a question yet, so please feel free to ask the comments, to use the comments to ask questions. We have some applauses, and I think people congratulating you for this fantastic talk. I would maybe start with a question. Yes, I'll probably start with the question related to the way that you've showed this journey of yours, and I want to know what's your next stage, and what comes for you in the future.
Yeah, thank you. Well, I think, the next stage is, we're generating preliminary data for this project as well. And I'm going on to the job market soon. So I'll probably be applying for faculty positions in the next year or so. And yeah, so I'm excited to see how to create this research program that I'm interested in.
Awesome. Thank you so much. We have a lot of congratulations that are coming through the chat. I think your talk was really exciting and impressive. For the sake of time, I will probably go to the next speaker now. And so thank you so much for this fantastic presentation.
Thank you.
All right, so for the next speaker, we will go back to Dr. Elijah Kolawole Oladipo that we should have back now if the connection is working. Let's see. Hi, Dr. Elijah. Can you, Kolawole Oladipo, can you see me or hear me? We can't see your video right now, but
I can see you.
Okay, I can hear you. We can't see you.
I can hear you. I can see you. I can see the two of you.
Okay. Can you share your slides? We don't see your video, but if you can share your slides, I think we should be able to.
Okay, let me share my slide. I can see the three of you.
We can see you now.
Okay, great.
And can we see your slides now?
Yep, perfect.
Awesome. Do you want to go to full screen mode?
Yep. Let me
All
share
right.
that.
I'm glad that we were able to have you connected back after a problem of connection. So just to go back to your introduction, you're an international fellow. You're presently holding a faculty position at the Department of Microbiology at Adekele University in Nigeria, and you focus on vaccine and diagnostic kits development for neglected tropical diseases. The floor is yours.
Yeah, good day. I really thank the organizer of this program for giving me the privilege to have some of our work discussed on the platform. Now, I'll be discussing about a topic on COVID-19 vaccine research and development journey in Africa. Now, vaccines is a product designed to help prevent diseases, and vaccine connect individuals from getting sick, such as chickenpox, flu, polio, hepatitis, measles, and the likes. Now, vaccines has been an effective public health tools, which have shown a lot of successful measures in saving millions against different form of diseases. Now, we have some challenges with vaccine. Race and ethnicity or geographical ancestry have also been shown to affect vaccine responsiveness. So the way we respond to vaccines differs from different geographical locations. Now we have an example of rotavirus vaccine's efficacy, which is considerably lower in Africa as compared to Europe and North America. And the lower in the efficacy has been attributed to host genomics, and that particularly involved in genes on the immune response and to the receptors and ligands. Now, what's the way out? We all had the pandemic issue like last year, and we thought that we should try our home best and move on and have a vaccine being developed from our own hand here too, and we focus on the use of spike glycoproteins. So then we were considering Africa countries, and these are the countries that we lay our hands on, as at the time we have isolate from Nigeria, Senegal, Egypt, Ghana, Tunisia, South Africa, Gambia, the Republic of Congo, and even the Wuhan, who was the reference isolate for this work. So as at the time of our work, we work on 60 to 80% of the isolate from these countries. And we're able to have about 1,416 B cells and 224 cytotoxic T lymphocytes, and we have 40 C B cells. And therefore, we had to construct a multiple epitope vaccine, and we added our adjuvant to it because protein subunit vaccine has its limitation, and once the limitation is immunogenicity, and for the immunogenicity to be increased, we have to add an adjuvant to it. So the vaccine eventually was not allergic and was highly antigenic. These are just some of the immune simulation that we did to see how the vaccine constructor is going to respond when it gets human, see the IgG and IgM, how it was produced by this. And we have the total T cell populations, how it then is that the CD4 T EPA lympho cells and the memory cell count. We have the T EPA cells, how it's where we have the natural killer cells that can kill the cell infected with the virus already. And these are two publications that we pushed out from the work from the developmental stages of the constructed vaccine. At the end of the day, our protein subunit vaccine made it on the landscape of novel coronavirus candidates developed by, collected by WHO. So making our team and that of Egypt the only team in Africa that made it to WHO list. And from there, we needed to synthesize our protein expression and scale up because we are working in Nigeria, in Oyo State, in Ugumosho, particularly in the lab that I founded by myself that I call a Helix Biogen Institute. So we could not synthesize based on some facilities on ground. So we have to get it across done in US by GenScript. And we tried two vectors. We did E. coli and we did insects. And this is just the expression of a protein, how the protein came out and every other thing. From there, we needed to proceed to the preclinical studies in order to evaluate the safety and the immunogenicity of the vaccine. So we have in our proposal that we submitted for ethical approval. We're considering mice and monkeys for a start based on the fund that we have. And the mice was conducted at the Elix Bajen Institute that I founded in Wumushu, Nigeria, whereby we enter into collaboration with a primate research institute at Nairobi, Kenya, for the monkey study. The monkey study is about starting because this study is an ongoing study. It's just ethical approval that was gotten from Ministry of Health in Oyo State, Nigeria here. And this is one of the results that was obtained for hematology of the mice that was done to see how safe, firstly, how safe the protein subunit vaccine is in animals. And we can see that here, basically we have the control, we have the single, and we have the booster. The animals and the booster got their booster after third week of the first shot. We have the MC, that mean composite volume, to see how it goes. I just have some selected results to show us here because the study is ongoing. So we have the vaccine on RBC. You can see for the control, we can see for the single, we can see for the booster, and we have for the weight of the animals, like the single week and the booster for the week. And we discovered that from people that have been vaccinated in Nigeria too, we discovered that even most people eat more when they got their second shot, and we are seeing the same thing in the animals. So this is just the prototype of the mechanism of action of the constructs that we design, how we use spike glycoprotein, added a adjuvant, presented to antigen-presenting cells, the helper cells, which is cell-mediated immunity, and the cytotoxic cell destroyed the cell infected by virus, and we have the infectious cells, both the B cells and T cells, remain in the body after infection to produce immunological memory. And we have the B cells, which have specific antibodies against SARS-CoV-2 made by B cells. So we have our directions. We've been able to complete the animal trial using mice. We've gotten the ethical approval. We are working on industrial scale-up to see some company to partner with in India for scale-up, because our country currently does not have the scale-up. So from there, we're going to start the clinical trial phase 1 after we have gotten approval from relevant agencies. All this work is on the pipeline. I have picture of fantastic young guys that are working in the lab with us, and because there's something we inculcated into our work as we are doing it, we call it a capacity building. We have young graduates in the countries from different institutions that are part of this team that we are training up, so that they also we have know about the technology so far. Then what are our future plans for this work is to get more grants, is to get more collaborations, and to move ahead. I want to thank my team members from different universities that have taken part in this work. Then I will thank my key members that have been very active moving on this work. So thank you for listening. This study is an ongoing study. Thank you.
Thank you, Dr. Elijah Kolawole Oladipo, for this talk. I'm really impressed to see the work that has been done, how you've been really going international to develop this vaccine. I think we're going to take some questions from the chat. I'll ask maybe a first question regarding, in the facilities that you're working in, with the tools that you have. What has been the most challenging aspects of putting together this study and this development?
Okay, I think the most challenges I've ever had is the funding, because the institute I had is a consultant firm. So most of the profit that we need, we channel into the development of the vaccine. So, and that has been the most difficult thing we have ever came across, because we know that once we are collaborating, if there are other things we cannot do, we have other scientists that can do that for us, and we form a national team. So in terms of expertise, we are improving and we are getting there. Like I said, the major thing that I've been happy about is that we have capacity that we are developing so that they can respond to such future pandemic.
Awesome. Thank you for this answer. I would say that if someone, I think we can contact you directly, or if there is someone else that should be contacted for collaborations, just let us know. And I think there will be probably questions for you in the chat, and hopefully we can have some connections going on to have some collaboration going on and some funding going on for you.
Okay.
I have a question from Medina in the chat. Very interesting to hear your progress on the vaccine. How do you think the different COVID variants may impact your vaccine?
Yeah, I think that is one of the factors that made us to start this work. I work on influenza virus for my master's and my PhD work, and I know that virus vaccine has been updated. And we felt that if we have this technology, as we have new variants involving, it's more easier for us to develop our vaccine and to meet the demand. You can see that currently now Africa is suffering from the availability of vaccine. So if we can do that, we will not need to wait for the Western world before we solve our problem by ourselves. Though we may need helping hand from them, but not totally depending on them before we solve the problem. And I want to tell you that the last in-house analysis that we did, our vaccine construct had 70% coverage of the Delta variant. So which shows that it's promising that we have a level of effectiveness against the variant that is coming up. And that is why we walk around all those countries in order to have a conserved region that is common to all of them, so that in case we have variant, they're able to handle them effectively.
Amazing. Thank you very much. I have another question from the chat. We have Ore Francis asking, saying great talk, and then sorry if I missed it, but do you think differences in post-translational modifications of protein vaccine could affect the long-term responses?
Yeah, there's likelihood of that, and that is why we are working on two different vectors. And when working on translational research, it's different from bench research. Like I said, we searching for E. coli and insect, and E. coli has a challenge that is something we call endotoxin. By the time you finish the expression, you need to remove your endotoxin. So if the endotoxin is removed and the quantity left is small, it will be good for post-production. And that is why, and we had problem with protein folding. And that's why we shifted to insect, where insect give us a better result than E. coli. The only challenge you have is that the generation time for E. coli is faster than that of insect. But we have a way to overcome these challenges.
Awesome. Thank you so much. I think we don't have any more questions in the chat, and they might come later. So I'll encourage you to look in the chat to see if there are more questions for you. Thank you so much, Dr. Elijah Kolawole Oladipo. And I'll go now to the next speaker. I'm very excited to have Dr. Cornelius Taabazuing joining us today. And Dr. Taabazuing is a postdoctoral fellow in the Department of Chemical Biology at Memorial Sloan Kettering Cancer Center in the lab of Dr. Daniel Bachovich. Sorry,
Bachovich.
Okay, sorry about that. He earned his bachelor's and his PhD at the University of Massachusetts. As a graduate student, he worked on understanding the chemistry of the enzymes that sense and regulate the adaptive response to hypoxia. His current research is focused on understanding the molecular mechanisms of innate immune activation and regulation. He recently discovered a new inflammasome belonging to the ZU5 domain, containing a family of proteins that activates the innate immune system. He uncovered the molecular mechanisms of activation and revealed the fundamental bidirectional crosstalk between apoptotic and pyroptotic signaling networks. As an independent investigator, he will study the molecular regulation of the other ZU5 proteins that sense genotoxic stress and regulate immunity. Dr. Taabazuing also cares deeply about improving diversity and equity in STEM and has been involved in many initiatives that are aimed at increasing representation. Thank you so much for being here, and I'm excited for your talk.
Thank you, Joel. And thank you to the organizers and to all the people out there on all the different platforms that have been uplifting our voices. I think it's really cool that this initiative has been able to span different states and even different continents, as we just saw. And it's really an honor to sort of close out this session, which has been full of phenomenal talks, and to tell you a little bit about the work that I've been doing to try to understand the regulation of the innate immune system. There are a couple of different sort of arms of the innate immune system, for example, the toll-like receptors and non-canonical inflammasomes. But for the purposes of this short talk, what I'm going to focus on is the canonical inflammasomes. And what I'm showing you here is just a subset of some of the more well-studied ones. And what you can appreciate is that they sense diverse yet specific insults. And the particular one that we study in the lab are these two inflammasomes called NLRP1 and CARD8. And the agent that we use to trigger their activation is this small molecule called Velbro-Pro. But very recently, pathogen proteases as well as pathogen RNA have been reported to activate these inflammasomes as well. The way that canonical inflammasomes are generally activated is that you have to have a danger signal that makes it inside the cytoplasm. Once that danger signal is intracellular, it can be either from the host or it can be from a pathogen. We have dedicated sensor proteins or pattern recognition receptors that'll sense that danger signal. They will then recruit this adapter protein called ASC, and the job of ASC is to recruit the pro-caspase-1 zymogen to the signaling platform. Once caspase-1 is there, it dimerizes, it gains activity towards itself, and then it's an enzyme that acts like a scissor, so it'll cleave things, and so it'll undergo autoproteolysis to generate the active caspase-1 species. Once caspase-1 is active, it can then cleave a number of different substrates, including the interleukin family of cytokines, as well as this protein called gastromin D. Normally, the C terminus of gastromin D is inhibitory and it represses the N terminus. But once caspase-1 cleaves gastromin D, that N terminus is released and it oligomerizes and then forms holes in the plasma membrane of the cells. And so this allows the diffusion of molecules that are small enough to pass through that hole. And it eventually leads to a lytic type of cell death called pyroptosis, or fiery cell death, essentially. And so this is how the innate immune system works. And gastromin D, well, canonical inflammasome activation, more specifically. And gastromin D is a fairly recent discovery. It was discovered in 2015. And when I started in 2016, some of the studies suggested that if you didn't have gastromin D, you still underwent cell death, but it was unclear what type of cell death. And it suggested that there might be other pyroptotic substrates. And so the first question I tried to tackle in the Bachochin lab was to answer the question of whether or not there were other pyroptotic substrates. And to do that, what we did was we knocked out gastromin D and then characterized the cell death that was happening. And so for this slide, what I want you to keep in mind is that we used this small molecule called Velbro-Pro to induce pyroptosis. And our readout for pyroptosis is going to be the cleavage of gastromin D into that P30 fragment, which induces pyroptosis. We also use this small molecule called etoposide, which induces apoptosis. And our readout for apoptosis is going to be processing of this protein called PARP. And so here I'm showing you a Western blot, which is looking at the proteins. And these are control cells, which have just a control guide RNA. And in our DMSO or control cells, we don't expect any biology to happen. And so that's exactly what we see. Nothing's happening. However, when we have VBP or our pyroptotic agent present, we expect gastromin D to be cleaved into that P30 fragment. And that's exactly what we see. When we have etoposide, which is our apoptosis-inducing agent, we expect to see PARP processing, and that's exactly what we see. But interestingly, we also saw alternative processing of gastromin D. So VBP activates caspase-1. So pyroptosis, VBP-mediated pyroptosis is absolutely dependent on caspase-1. So if you don't have caspase-1 present, you don't expect to see pyroptosis. And so here in our caspase-1 knockout cells, we have our pyroptotic agent, and we no longer see processing of gastromin D into that P30 fragment. However, if we have our apoptotic agent, we do see what's indicative of apoptosis, which is PARP processing. And again, we see that alternative splicing of gastromin D. And now, if we don't have gastromin D present, which is the pyroptotic substrate, what we see is that now with our pyroptotic agent or inducer, we now see PARP cleavage, and that's true for 2 different clones of the gastromin D knockout. And so this suggests that these cells are now undergoing apoptosis as opposed to pyroptosis. And we can further measure this because when cells die, the phospholipid membrane inverts, and there's a dye called annexin 5 that binds to that. And so in our control cells, which undergo pyroptosis, we don't see an increase in that annexin 5 signal, but in our gastromin D knockout cells, which biochemically are supposed to be undergoing apoptosis, we do in fact see an increase in that annexin 5 signal. And to cut a long story short, it's essentially what we discovered was that if you have a pyroptotic agent, in our case, we're using Available you'll activate caspase 1. Caspase 1 will cleave gastromin D, and this will induce pyroptosis. And as you can see, the morphology of pyroptosis is quite distinct. And so these cells essentially just burst open. So this one will pop open, boom, and then this one will pop next. So it's quite distinct. However, what we discovered was if you do not have gastromin D, caspase 1 will then activate caspase 3 and 7, which will then go on to induce apoptosis. And so this is really cool because it suggests that if a cell has a pyroptotic signal, it's committed to undergoing cell death, whether via an inflammatory phenotype or what we typically think of as an immunologically silent phenotype. But what was even more interesting was that during the course of apoptosis, the activated caspase 3 and 7 enzymes also process gastromin D at an alternative site. And that was that alternative processing that we were seeing with our apoptosis inducer. And when this happens, it abolishes the pyroptotic function of gastromin D. And so there's bidirectional regulation here where caspase 1 can feed into the apoptotic pathway and the apoptotic pathway feeds into the pyroptotic pathway. And the morphology for apoptotic cells is very distinct from pyroptosis, and we're able to see the differences here. And so you can see that apoptotic cells sort of undergo membrane blepping and form these sort of scraggly looking cells. And so we discovered this bidirectional crosstalk, but in order to induce pyroptosis, it's usually mediated by the pattern recognition receptors. And so we next asked what pattern recognition receptor mediates VBP-induced pyroptosis. And we had sort of narrowed it down to a couple of PRRs, and we found out that in mouse, the pattern recognition receptor that mediates VBP-induced pyroptosis is NLRP1B and also NLRP1A. And so in humans, we have NLRP1, and this is the domain sort of schematic of these proteins. And you see that the human one differs from the mouse one by the addition of this extra pyrin domain. And so we thought that in humans, it's going to be NLRP1 as well. And if that's true, one would predict that if this is important for VBP-mediated cell death and pyroptosis, if you knock it out, you would no longer see a response. They would no longer be sensitive to VBP. And so I knocked out NLRP1 in monocytes, and here we're looking at LDH release. So when cells die, they release lactate dehydrogenase. And what you see is that in the control cells, they're responsive to VBP, and in the NLRP1 knockout cells, they're still responsive to VBP. And so in humans, NLRP1 does not seem to be the inflammasome that mediates VBP-induced pyroptosis. However, humans have this additional protein called CARD8, which doesn't exist in mice. And so we thought that this might be the putative inflammasome that was previously unknown. And what we see is that if we knock out CARD8, these cells are no longer sensitive to VBP. And so CARD8 seems to be the inflammasome that's mediating pyroptosis in response to VBP in these monocytic cell lines. And if you look at the Western blot, you indeed see gastromin D processing, which is indicative of pyroptosis. But curiously, we saw that the N-terminus seemed to disappear. And again, due to time constraints, I'm going to cut a long story short and sort of summarize, a year's worth of work that we've done to sort of uncover the molecular mechanism of these inflammasomes. And this was a truly collaborative effort with a lot of what you can see is a very diverse and awesome group of researchers. And I'm hoping that one day I can emulate sort of the lab culture that we have, where we can have a diverse collaborative group working together. And so together, what we found was that in response to VBP, it inhibits these serine hydrolases and it'll activate these inflammasomes. We also found that it can induce the human inflammasome in different cell types. But what's unique about these inflammasomes compared to the other ones is that they have this ZU5 domain, which allows them to undergo autoproteolysis. So this is a post-translational autoproteolysis. And so you essentially generate two different halves of the protein, the N-terminal half and the C-terminal half. And the C-terminal half is sort of like the warhead portion of this, if you would liken it to a missile or something. So it's the business end. And so because these two halves are non-covalently associated, when you activate this inflammasome, the N-terminus gets degraded by the proteasome and it releases the C-terminus, which then forms the inflammasome, which recruits caspase-1 and then activates it. But it wasn't clear how caspase-1 gets activated on the inflammasome. So that was a question we next tried to address. And one of the sort of paradigms in the caspase-1 field that existed for about a decade was that the pro form of caspase-1 was active. And the reason why this existed was because about a decade ago, they took the mouse pro-caspase-1 zymogen, and one of the hallmarks of caspase-1 activation is its processing. And they mutated the putative processing site. So here, the human, there are 6, but in the mice, there's an additional one here in this interdomain linker region. And this is the catalytic cysteine. And when they mutated all the processing sites, they saw cell death, but they didn't see processing of caspase-1. And so that's how it got established that the pro form of caspase-1 was active. And so we examined this a little bit further. And here, what we did was we took the human caspase-1 and we also mutated the putative processing sites. So here, if you mutate aspartic acid 297, you prevent processing at this site, but you can still process at the other sites. And so we made these respective mutations. And what we did next was we took monocytes that were null for caspase-1, so Th1 caspase-1 knockout cells, and we reintroduced these different variants of caspase-1 back into the cells. And so here are our control cells. What you see is that if you treat them with available pro, you expect to see gastrin D processing into that P30 band, indicative of pyroptosis, and that's exactly what we see. If you have caspase-1 knockout cell lines expressing GFP, you don't expect to see any pyroptosis, and we don't see gastrin D processing. If you put back wild-type caspase-1, you see that there's some aberrant activation, but it's further enhanced by VBP. And what's interesting is if you have any of the interdomain linker regions mutated, so it's not able to fully process there, you see attenuated gastrin D processing or attenuated pyroptosis. And very interestingly, if you don't cleave the interdomain linker altogether, you see that this enzyme is essentially dead, for lack of a better word. It can't undergo pyroptosis. And as expected, this is a catalytic cysteine inactivation, and we don't see any pyroptosis there. And also, of course, if the interdomain is not inactive, then you would expect that the fully uncleavable form is inactive. And we also went back and verified this in the mouse and found that it's a dysregulated enzyme if you mutate this processing site, and you get cell death, but it's non-pyroptotic cell death, and that interdomain linker processing was similarly required for activation of the mouse variant. And so this sort of just summarizes what I essentially just told you, is that if you can process caspase-1 at all the putative sites, you get full activation. If you inhibit processing at any of the interdomain linker regions, you get attenuated activation. And if you can't process that interdomain linker, you get complete inactivation. And to sort of summarize everything that I've learned or contributed to during my posa is that we have available pro, which inhibits these serine hydrolases. And as a consequence of their inhibition, you can either activate the NLRP1 or CARD8 inflammasome. And the mechanism for their activation is N-terminal degradation. However, there are distinctions. For NLRP1, you require the adapter molecule ASC to nucleate caspase-1, which can then mediate that interdomain linker processing, and then it'll go on to cleave substrates, which include the cytokines as well as gastrin D. However, CARD8 does not use ASC. It nucleates caspase-1 directly to mediate that interdomain linker processing. and it can then process gastromin D and induce pyroptosis. And since this is the sort of translational portion of it, I thought I would sort of tell you some of the more translational aspects or potential of uh, the induction of pyroptosis. And we in the lab have done this leukemia model where we saw that using these DPPA9 inhibitors, it's a potentially viable strategy for treating cancers. And there's a lot of ongoing work in this region. And it's not just humans that can, inactivate gastromin D. There are some viruses that can cleave gastromin D at alternative sites to evade the immune system. So understanding this mechanism might help us understand immunoevasive viruses as well. And for autoinflammatory disease, there are mutations that are in NLRP1, germline mutations that cause autoactivation and lead to skin lesions, which predisposes these people to cancer. And so Darren is a graduate student who was very talented, and he has since moved on. But here's the MV4 leukemia model he did. He engrafted these mice with MV4 cells, and you can see that once they have a high burden, if you treat with Valbol or this DPPA9 inhibitor, you see that there's a dramatic reduction in the cancer burden. And you can quantify this where VBP essentially stops the cancer progression, whereas the control treatment, they progress. And essentially what this translates to is a 95% reduction in the tumor burden, and it leads to a 25-day survival advantage. And this is a hit that I just got from in PubMed this week, where there's a company that's using these DPP inhibitors in combination with anti-PD-1 antibodies. And so this is an ongoing area of research, and a lot of people are interested in using pyroptosis in this manner. And with that, this has been, again, an incredibly supportive and collaborative environment to do science in, and I'm really grateful for it. I'd like to thank all the lab members, the past and the present, as well as the Black and Immuno organizers. And again, all of you for listening, and all the people out there uplifting our voices. It's truly appreciated. And with that, I will take any questions.
Awesome. Thank you so much, Dr. Cornelius Taabazuing, for this great talk dissecting the pathways that lead to pyroptosis. We have the time for a few questions. We have two in the chat already. Great work from Catherine Mayer-Barber. Do you think that aborted redirected pyroptosis in GAS-D, KO, or CARA8 ultimately leads to cytolysis via GAS-DE downstream of Cas+3 if you wait long enough after apoptosis induction? It's a very specific question.
Yeah, that's because caspase-3 has been shown to cleave gastromin E, which then mediates pyroptosis. So there's a really intricate sort of plastic network of cells. And I would say that it depends. If the cell is expressing gastromin E, then most likely it's possible that the activation of caspase-3 will lead to gastromin E-mediated pyroptosis. So that would be if the cell is expressing it. But if it's not, you would probably still get apoptosis. But if all the components are there, it's likely that kinetically pyroptosis is so much faster that you're going to get gastromin D, caspase-1 mediated gastromin D cleavage before you get to caspase-3 mediated gastromin E cleavage.
Awesome. Thank you for this answer. We have a question from Chris Bourne. Great job, Cornelius. My question is related to Catherine's. In the absence of gasdermin D, is the apoptotic cell death canonical and still immunologically silent, or are there other factors that would still contribute to inflammation in that context?
Yeah, that's a good question. Because, I mentioned that we typically think of apoptotic cell death as immunologically silent. In the context of, the absence of gastromin D, one would think so, because you probably would not be cleaving cytokines actually you would, but you wouldn't be releasing them in the same way. But eventually you get secondary necrosis with apoptosis if you wait long enough. And so in that manner, it might still be immunogenic, because if you wait long enough, you will get cellular rupture. And so it's a good question. I would say that it depends on when you look, ultimately, whether or not you would characterize it as immunologically silent or if it's still inflammatory. But as long as you are, if you get caspase-1 activation and you can still process cytokines, and the cell loses the cellular integrity in some way, you would eventually get immunogenic cell death, or immunologically active, if you will.
Awesome. We still have a few minutes for an additional question, so feel free to ask in the chat. In the meantime, I'm going to ask you, Cornelius, you're opening your lab, so I'm assuming that you're recruiting.
Yes, I am. I'm looking for tags. Hopefully, graduate students will join once I start, and also a postdoc. So, if you're interested in mechanisms of cell death, please reach out to me. I think my email can be found somewhere, or rather, if not, just use social media or something and let me know. I'm very interested in recruiting a postdoc and a tech in the next month or two, hopefully, because we open up in January.
Perfect. This is awesome. Yes, I think people can find you on Twitter. I think you're very present. I've seen you a lot over there. And yes,
yeah,
if there is
amazing and surreal how much people have been helping to uplift our voices on that platform. So I truly appreciate it.
Absolutely. And if someone is interested and cannot find, the contact of Cornelius, please shoot an email to Black in Immuno, or a message, and we'll get people in contact for sure. All right. So we have a lot of applause in the different in the chat. Sorry, and I think someone just dropped your handle. Your Twitter handle
Oh,
is
perfect.
at Tabaman, right?
Yes. Yes.
All right, and yes, and with that, we are going to close this session. Thank you very much for all the speakers for these fantastic talks. I personally have been very impressed by the breadth of this fantastic research that is being done. And I'm very excited to see how this work is going to continue in the future. We'll be definitely following all these amazing speakers. Thank you, everyone, for attending this session. And we'll have a panel that is going to start in about 15 minutes now. And so you will all invited to join this panel. And we hope that everyone continues to have a fantastic Black in Immuno week. Thank you for joining today, and I'll see you on the next event. Thank you very much.